Hi, my name is Jelena Skorek, and the topic of my presentation is ion channels and neuronal communication. Within this topic, I will define what neural communication is. I'll talk about different membrane potentials, ion channels involved in changes of membrane potentials, graded potential, action potential, about neurons and different synapses. So what is neuronal communication? Neuronal communication is one of two major regulatory systems of the body that help ensure life-sustaining coordinated responses. The other one is endocrine system, which will not be part of today's presentation. Neuronal communication is accomplished by means of nerve cells or neurons, which are specialized for rapid electrical signaling and for secreting transmitters, which are short-distance chemical messengers that act on nearby target organs. The nervous system exerts rapid control over most of the body's muscles and exocrine secretions. But before I can talk any further about mechanism of neuronal communication, I need to define membrane potential, graded potential. All body cells display a membrane potential, which is separation of positive and negative charges across the membrane. This potential is related to the uneven distribution of sodium and potassium ions and large intracellular protein anions between the intracellular fluid and the extracellular fluid, and to the different permeability of the plasma membrane to these ions. As you can see on this picture, concentration of sodium is higher out of the cell and concentration of potassium is higher in the cell. Also inside the cell, there are negatively charged protein ions which cannot exit the cell. These concentration differences are maintained by the sodium-potassium pump at the expense of energy. In addition to the active carrier mechanism, sodium and potassium can passively cross the membrane through protein channels specific for them. It is usually much easier for potassium than for sodium to move across the membrane because membrane is 25 to 30 times more permeable to potassium than sodium. The sodium-potassium pump transports 3 sodium ions out of the cell for every 2 potassium ions it transports in. Because sodium and potassium are both positive ions, this unequal transport separates charges across the membrane, with the outside becoming relatively more positive and the inside becoming relatively more negative, as more positive ions are transported out than in. However, this active transport mechanism only separates enough charges to generate a small membrane potential. Most of the membrane potential results from the passive diffusion of potassium and sodium down concentration gradients through leak channels, which I will be discussing a little bit more about on the one of upcoming slides. At resting, the membrane is polarized at -70 millivolts in a typical neuron, and this is called resting potential. On the picture on this slide, in the middle of the graph, we have a value of -70, which represent resting membrane potential. During depolarization, which is caused by cell excitation, the membrane becomes less polarized or less negative than in resting potential, with potential moving closer to 0 millivolts. During depolarization, potential can become even positive during action potential, which I will talk about a little bit later in this presentation. After depolarization, membrane returns to a resting potential, and this is called repolarization. During repolarization, membrane can become more polarized or more negative inside the cell, which is called hyperpolarization. Changes in membrane potential are brought about by changes in ion movement across the membrane. So if we remember from the previous slide that net inward flow of positively charged ions increases compared to the resting state, such as more sodium moves in the membrane, in the cell, causing membrane to depolarize and become less negative inside. By contrast, if the net outward flow of positively charged ions increases compared to the resting state, such as more potassium moves out the membrane, hyperpolarization will occur, which means it becomes more negative inside. Changes in ion movement are brought about by changes in the membrane permeability in response to triggering events. A triggering event triggers a change in membrane potential by altering membrane permeability and consequently altering ion flow across the membrane. These ion movements redistribute charge, which changes the potential. The question is, how do these ions move across the membrane? Well, all these ions we mentioned are water-soluble and cannot penetrate the plasma membrane's lipid bilayer. They can cross the membrane only through channels specific for them or by carrier-mediated transport. Membrane channels, which I mentioned earlier when I talked about membrane potential, are specific membrane embedded proteins which have hydrophobic ends attached to the lipid bilayer and hydrophilic part that creates the inside of the channel through which ions can pass through. These channels can be either leak channels or gated channels. Leak channels are open all the time, permitting unregulated leakage of their specific ion across the membrane through the channels. The most important one from this group are sodium and potassium leak channels. Gated channels, in contrast, have gates that can be open or closed, permitting ion passage through the channels when open, in preventing ion passage through the channels when closed. Gate opening and closing occurs in response to a triggering event that causes a change in the conformation or shape of the protein that forms the gated channel. There are 4 kinds of gated channels depending on the factor that causes the channel to change shape. On this picture, at the right bottom corner, we have voltage gated channels, which open or close in response to changes in membrane potential, which I'm going to describe a little bit more in next slide. Then we have chemically gated channels that change shape in response to binding of a specific extracellular chemical messenger. Then we have mechanical gated channels above the voltage gated channels that change shape in response to stretching of membrane or other mechanical deformation. And finally, we have thermally gated channels which respond to local changes in temperature, which are cold or heat. So I said that voltage-gated channels open or close in response to changes in membrane potential. For this topic, the most important voltage channels are sodium and potassium one. Sodium-potassium channels have 2 gates that are shown on this first picture on the slide. The activation gate guards the inside of the channel by opening and closing like a sliding door. The other door is called inactivation gate and consists of a ball and chain-like sequence of amino acids at the channel opening facing the intracellular fluid, as you can see here. This gate is open when the ball is hanging free on the end of the chain and closed when the ball binds to the channel opening and blocking the opening, like on the third picture here. Both gates must be open to permit passage of sodium. This voltage-gated sodium channel can exist in three conformations. The first one, and it's shown here on the picture, is closed but capable of opening. The second one is open or activated, and we can see ion channels, sodium ions going through the channel. And the third one is closed and not capable of opening or inactivated. The channel moves through these various conformations as a result of voltage changes that take place during the action potential, which we will discuss shortly. Another important voltage-gated channel is potassium one, and potassium channel is a lot simpler than the sodium, and it has only one activation gate, which can be either open or closed. As I said earlier, changes in ion concentrations between intracellular and extracellular fluid as a result of their movement through the membrane cause producing electrical signals. There are two basic electrical signals, graded potential, which serves as a short distance signal, and action potential, which signal over long distances. Graded potentials are local changes in membrane potential, and they vary in their strength. For example, membrane potential could change from -70 to -60 millivolts, which is 10 millivolt graded potential, or from 70 to 50 millivolts, which is 20 millivolt graded potential. Graded potentials are usually produced by specific triggering event, which causes voltage-gated channels to open at a specific region of the excitable cell membrane. The resultant ion movement produces the graded potential, which most commonly is depolarization resulting from net sodium entry. So if you look at this picture, the first one shows more sodium ions out of the cell, and so the inside is more negative. After triggering event on the second picture, voltage-gated sodium channels open and sodium enters the cell, so the inside becomes less negative, producing the graded potential. The graded potential is confined to this small space, and it's called active area. The neighboring areas are still at resting potential. Outside the cell, the active area is relatively less positive than adjacent inactive areas. Because of this difference in potential, charges carried by ions passively flow between the active and inactive regions on both the inside and the outside of a membrane, producing a current. Current carried by ions can move across the membrane only through these ion channels. Although graded potential have limited signaling distance, it is very important to the body's function. Graded potentials can initiate action potentials, which is the long distance signaling. So besides graded potential, we have action potential, which is brief, rapid, large change in membrane potential during which the potential actually reverses so that the inside of the excitable cell rapidly becomes more positive than the outside. The strength of action potential does not diminish as it travels from their site of initiation throughout the remainder of the cell membrane, And that is why action potential serves as a long-distance signaling. If you look at the picture, picture number 1, at resting potential of negative 70 millivolts, all voltage-gated channels for both sodium and potassium are closed. Therefore, sodium and potassium cannot pass through these voltage-gated channels at resting potential. After the membrane is stimulated, at the picture number 2, gated channels open and the cell goes through depolarization. The new region of the membrane starts to depolarize toward the threshold, which is the critical level of membrane potential, typically between negative 50 and negative 55 millivolts. This depolarization causes the activation gates of some voltage-gated sodium channels in the new region to open, and sodium starts to move in. The inward movement of positively charged sodium depolarizes the membrane further, opening even more voltage-gated sodium channels and allowing more sodium to enter, and so on in positive feedback cycle. So at this graph, the number 1 depicts the picture number 1, resting potential. Threshold shows a rising of the membrane's potential because the sodium ions are entering, and then there's explosive opening of these gated channels and explosive entering of sodium ions, which brings membrane potential to the positive level of 30 millivolts. At the peak of action potential here, the potential does not become any more positive because sodium channel starts to close. With inactivation of sodium channels, the voltage-gated potassium channels start to slowly open, and the membrane potential falls down to the level of resting potential. So we have 3 events that occur here. The first one is rapid opening of sodium activation gates, which permits sodium to enter the cell. The second one is slow closing of sodium inactivation gates, which stops further sodium entry after a brief time delay, which keeps the potential from rising any further. And third event is slow opening of potassium gates, which is in large part of responsible for potential plummeting from its peak back to resting. The membrane potential gradually returns to resting after closure of sodium channels as potassium continue to leak out, but no further sodium enter. The outward movement of potassium rapidly restores the negative resting potential. These potassium channels are slow close, which causes membrane at the end of repolarization to become a little bit more negative, which is called hyperpolarization. Now, when we know more about membrane potentials and all the channels involved in its changes, we can talk about specific mechanism of neuronal communication. But before, I need to define main parts of the neuronal cell. Those are cell body, the dendrites, and the axon. Dendrites are shorter extensions that receive impulses from the other neurons, and axon is the longer extension of the neuronal cell, which carries impulses from the body away toward the other neuronal cell or the other organ. Axons vary in length from less than a millimeter in neurons that communicate only with neighboring cells to longer than a meter in the neurons that communicate with distant parts of the nervous system or peripheral organs. The first portion of the axon plus the region of the cell body from which the axon leaves are known as axon, hillock, or initial segment. This is the neuron's triggering zone that contains large amount of voltage-gated sodium channels. Once an action potential is initiated at the axon hillock, no further triggering event is necessary to activate the remainder of the nerve fiber. The impulse is automatically conducted through the neuron, and this is called contiguous conduction, which involves spread of the action potential along every patch of membrane down the length of the axon. When an action potential reaches the axon terminals, that's the ending of the axon, they release a chemical messenger that alters the activity of the cell on which the neuron terminates. A neuron may terminate on one of three structures: a muscle, a gland, or another neuron. Therefore, depending on where a neuron terminates, it can cause a muscle to contract, a gland cell to secrete, another neuron to convey an electrical message along a nerve pathway, or some other function. When a neuron terminates on a muscle or a gland, the neuron is said to innervate or supply the structure. The junction between two neurons is called synapse. And there are two types of synapses. We have electrical synapses and chemical synapses, depending on how information is transferred between the two neurons. In an electrical synapses, two neurons are connected by gap junctions, which allow charge-carrying ions to flow directly between the two cells in either direction. Although electrical synapses lead to unbroken transmission of electrical signals and are extremely rapid, this type of connection is essentially on or off and is unregulated. At an electrical synapse, an action potential in one neuron always leads to an action potential in the connected neuron. Electrical synapses are not as common as chemical synapses in the human nervous system, and they're mostly present in smooth muscles and cardiac muscle. Most synapses in the human nervous system are chemical synapses, at which a chemical messenger transmits information one way across a space separating the two neurons. A chemical synapse typically involves a junction between an axon terminal of the one neuron, known as presynaptic, and on this picture it's a yellow one, and the dendrites or cell body of the second neuron known as postsynaptic, which is green on this picture. The axon terminal of the presynaptic neuron, which conducts its action potentials toward the synapse, ends in a slight swelling, which is called synaptic knob. The synaptic knob contains synaptic vesicles, which store a specific chemical messenger called transmitter that has been synthesized and packaged by the presynaptic neuron. The space between the presynaptic and postsynaptic neuron is called synaptic cleft. So an action potential in the presynaptic neuron alters the postsynaptic neuron's potential by chemical means. Synapses operate in one direction only, so presynaptic neuron brings about changes in the membrane potential of the postsynaptic neuron, but the postsynaptic neuron does not directly influence the potential of the presynaptic neuron. When an action potential in a presynaptic neuron has been propagated to the axon terminal, This local change in potential triggers the opening of voltage-gated calcium channels in the synaptic knob. Calcium ions promote the release of neurotransmitter from some synaptic vesicles into synaptic cleft. Released neurotransmitter diffuses across the cleft and binds with specific protein receptor on the postsynaptic membrane. This binding of neurotransmitter to the receptor channels causes the channels to open, changing the ion permeability and thus the potential of the postsynaptic neuron. These are chemically gated channels in contrast to voltage-gated channels responsible for action potential and for calcium influx into the synaptic knob. Because the presynaptic terminal releases the neurotransmitter and the subsynaptic membrane of the postsynaptic neuron has receptor channels for the neurotransmitter, the synapse can operate only in one direction, from presynaptic to postsynaptic neuron. Different neurotransmitters cause different ion permeability changes, so depending on type of change they cause, there are two types of synapses: excitatory synapses and inhibitory synapses. An excitatory synapse contains receptor channels to which the neurotransmitter binds, and they are non-specific ion channels that permit simultaneous passage of sodium and potassium ions through them. Permeability change induced at an excitatory synapse results in the movement of a few potassium ions out of the postsynaptic neuron, while a large number of sodium ions simultaneously enter this neuron. The result is net movement of positive ions into the cell, which makes the inside of the cell slightly less negative, bringing the postsynaptic neuron's membrane closer to threshold, which means that the membrane is now more excitable than when at rest. Postsynaptic potential occurring at an excitatory synapse is called an excitatory postsynaptic potential, or EPSP. At an inhibitory synapse, binding of a neurotransmitter with its receptor channels increases the permeability of the subsynaptic membrane to either potassium or chloride ions depending on the synapse. The resulting ion movements bring about a small hyperpolarization of the postsynaptic neuron, which means greater internal negativity. In the case of increased potassium permeability, more positive charges leave the cell, leaving more negative charges behind on the inside. In the case of increased chloride permeability, because the concentration of chloride is higher in the extracellular fluid, negative charges enter the cell in the form of chloride ions. In either case, the small hyperpolarization moves the membrane potential even further from threshold, reducing the likelihood that the postsynaptic neuron will reach threshold and undergo an action potential. That would be it for this presentation. Thank you for your attention.